Understanding bacteria. Bacteria are single-cell microorganisms that are classified as prokaryotes. Unlike eukaryotic cells, they have membrane-bound nucleus and other organisms. Bacteria like nucleus and other membrane-bound structures. He said that genetic material is found in circular DNA molecules located in the nucleus region. Structure of bacteria. Bacteria are composed of rigid cell wall that is surrounded by a cell membrane. Bacteria have cytoplasm, which is a gel-like substance within the bacterial cell, where various cellular activities take place. It contains cell genetic material called DNA. Nucleus. Unlike eukaryotic cells, bacteria lack true nucleus. Instead, their genetic material is located in the region called nucleus. Ribosome. Bacterial ribosomes are responsible for protein synthesis. They read the genetic code from DNA and assemble amino acids into protein. Flagella. Some bacteria have flagella whip-like appendages that produce from the surface of the cell. Pili or fimbriae. These are hair-like structures that extend from the surface of bacteria. Pili are involved in various functions, including adherence to surface and transfer of genetic material during process of fungification. Capsule. Some bacteria have a protective layer called capsule, which is composed of polysaccharide. This capsule helps bacteria evade the host immune system and contribute to the bacterial ability to cause disease. Shape of bacteria. Bacteria exhibit various shapes, and their classification is based on their morphological characteristics. We have the cocci. Each are spherical and round bacteria. An example are Streptococcus and Streptococcus. Bacilli, which are rod-like bacteria that are cylindrical in shape. An example is E. coli and Bacillus anthracis. Spirilla. Spirilla-shaped bacteria with rigid helical structure. An example is Spirillum volutans. Spirochete. These are thin, flexible, spiral-shaped bacteria with distinctive corkscrew appearance. Example is Treponema pallidum. Vibrio. These are common-shaped bacteria and are curved or bent, broad-shaped. Example is Vibrio cholerae. Filamentous bacteria. These are elongated, thread-like bacteria with a filamentous structure. An example are Actinobacteria and Streptomyces. Coccobacilli. These are intermediate shaped between cocci and bacilli appearing some macova, and an example is Haemophilus influenzae. Ubiquity of bacteria. Bacteria are ubiquitous and can be found everywhere on Earth. They can be found in soil, water, and various natural habitats. They can be found in human hosts, and these are known as human microbiomes. And they inhibit different body sites such as skin, mouth, gastrointestinal tract, and reproductive organs, playing an essential role in maintaining health and supporting body functions. They can also live in extreme environments with conditions that are once thought to be inhabitable for life, such as acidic hot springs, deep sea hypothermal vents, and some extremophiles can survive in extreme temperature, pH level, and pressure. They also have airborne bacteria that are present in the air and contribute to the composition of atmospheric aerosols. They can also be found in food and are used in fermentation of food. Good and bad bacteria. Apart from the fact that some bacteria are pathogenic in nature, bad bacteria that causes disease, there are some good bacteria too that are used in the industrial processes. For example, the lactobacillus species are used in fermentation process in industry. Bacterial diseases in human. Bacteria are known to cause various infections in human, such bacterial meningitis, pneumonia, skin infection, urinary tract infections, gastritis, sinusitis, hair infection, and also sexually transmitted diseases. So there are beneficial bacteria and also pathogenic that cause these diseases. And there are some opportunistic pathogenic bacteria which are able to cause infection when the immune system of the host is down. Beneficial bacteria. An example of beneficial bacteria are bacteria involved in nitrogen cycle. They are involved in the nitrogen fixation. They are present mostly in the root nodules of legumes, and they are also known as nitrogen-fixing bacteria, which convert atmospheric nitrogen into ammonia or related compounds for easy assimilation by plants. We also have ammonification, which are bacteria that help in the decomposition of dead plant and animal and waste products, and are involved in producing ammonium as a result of their decomposition activities. We also have nitrifying bacteria, which help convert the ammonium into nitrate and then to nitrate, which is usable by plants in production of many important components such as proteins needed by plants for their food. We also have assimilation. And we also have detoxifying bacteria, which help to convert nitrate back to nitrates and then nitrates back to nitrogen gas, which are released back into the atmosphere, and thus the cycle continues. Bacteria also involved in carbon cycle, whereas they help in the decomposition of plants and animals whenever they die, therefore leading to the release of carbon into the soil and into the atmosphere. They are also involved in fossil fuel formation. For over millions of years, the remains of plants and animals can be transformed into fossil fuel like coal, oil, natural gas. With this fossil fuel, they are burnt into energy, and carbon is subsequently released into the atmosphere. Human microbiome. These are bacteria that are known to be resident in humans. Some are known to be resident in nose, mouth, lung, stomach, colons, also in sexual organs to maintain the pH and production to kill other infectious microorganisms. And also, they can be found in skins. Whenever they are found in skins, they practice what is called territorialism, where we are producing microbioactive elements that inhibit the growth of pathogenic organisms. Single cell production. Single cell protein refer to protein-rich biomass derived from cultivation of microorganisms. Typically unicellular microorganisms like bacteria, yeast, fungi, or algae. These microorganisms are cultivated to serve as source of proteins for human and animal consumption, and can be exploited as potential alternative protein source to address global food security and sustainability concerns. Microorganism has protein source. Various microorganisms can be used to produce single cell proteins. Common example of bacteria used is Spirilla, yeast, Saccharomyces cerevisiae. We also have fungi, Fusarium, Venetium, and microalgae. In the cultivation process, single-cell proteins are produced through a controlled cultivation process. Microorganisms are growing in bioreactor or fermenter tanks, and they are provided with nutrients including carbon source, nitrogen source, and other minerals to support their growth. Nutrient utilization. Microorganisms using single-cell proteins production can utilize unconventional and non-food sources as nutrients. This will sure make single-cell protein more sustainably compared to traditional protein source, and they may not compete directly with human resources. After the whole process, single-cell proteins are extracted and are used as food supplement. Microorganisms are also involved in water treatment. For example, bacteria, the aerobic bacteria. These are organisms that require organisms for their metabolism and play vital role in the oxidation of organic matter during water treatment. They help break down complex organic compounds to simpler form. We also have the aerobic bacteria functioning in the environment with limited oxygen. Aerobic bacteria contribute to processes like denitrification and decomposition of organic matter in sludge digestion. Maintaining a healthy relationship with bacteria. Practice good hygiene. Avoid overuse of antibiotics. Include prebiotics in your diet. Eat a diverse and balanced diet. Stay hydrated. Exercise regularly. Get sufficient sleep. Manage stress. Limited processed food and sugar. Maintain a healthy weight. Avoid unnecessary disinfectants. Consider prebiotics. Consult with healthcare professionals. Steps for food safety involve cleaning of hands, then preparation of food under hygienic conditions, making sure that your food is well cooked. Avoid eating raw food, and then if food had to be left over, leave them in refrigerator. Antibiotic stewardship. Antibiotic stewardship is a coordinated effort aimed at optimizing the use of antibiotics to ensure effective treatment of infections while minimizing the development of antibiotic resistance. This practice involves the set of strategy and policy implementation by healthcare professional institutions and public health organizations to promote responsible and judicious use of antibiotics. The goal of antibiotic stewardship include improving patient outcome, reducing the spread of antibiotic-resistant bacteria, and preserving the effectiveness of available antibiotics. Here are the key components of antibiotic stewardship. We have guideline and policy to appropriate the use of antibiotics and guide healthcare practitioners in prescribing antibiotics only when necessary. We also have education and training, which is provided for healthcare professionals, including physicians, nurses, and pharmacists, and is necessary for raising awareness of antibiotic resistance. Diagnostic testing. Encouraging the use of accurate and timely diagnostic tests help healthcare providers to confirm the presence of bacterial infection before prescription. Surveillance and mentoring. Implementing surveillance help to monitor antibiotic use and resistance patterns, help the healthcare institution identify trends, assess impact to stewardship intervention, and make informed decisions about antibiotic prescribing practices. De-escalation of therapy. When feasible, antibiotics stewardship promote the de-escalation of therapy. This involves reassessing antibiotics treatment based on clinical and laboratory data and narrowing of spectrum of antibiotics to minimize the impact of patient microbiome. During treatment, setting appropriate duration of antibiotic treatment help prevent unnecessary prolonged causes, reducing the risk of antibiotic resistance and minimizing the side effects of patient. Communication and patient education. Promoting effective communication between healthcare and patients is crucial. Educating patients about the appropriate use of antibiotics, the importance of completing prescribed antibiotics, and risk of association with misuse help foster a shared responsibility of stewardship. Antibiotic development. Supporting research and development efforts to discover new antibiotics and alternative treatment is essential for addressing emerging resistance challenges and ensuring a sustainable arsenal for effective antimicrobial agents. Thanks for listening. Once again, I'm Samuel from ATG Ventures. Thanks for watching today's episode of BioNuggets on the topic Understand the Bacteria. Please do well to subscribe to this channel and turn on the notification button so that you'll be the first person to receive awesome videos like this when I release one. See you next time.